Hello, 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 everybody. Welcome. Welcome to the stream, everybody. Welcome. Hello, hello. Quite a bit of you here today to hear me talk about the obelisk. I'll start off stream by just saying hello to everybody, and then I will introduce our very special guest here. Hello, Hero. Hello, Either. Hello, Gogo. Hello, Polski. Hello, Yishu. Hello, Lazard. Hello, The Texture Pack. Hello, Sarah. Hello, Shadower, and hello, Dreamless. Hello, Infi. Hello, Jacob. Hello, Bootme. Hello, Kaizen. Hello, Surprise Pants. Hello, Tears Near. Hello, The Troll. Hello. Hello, hello. Hello, Joel and The Way. Hello, all the new subscribers. Hey, Penboo. Welcome, welcome. Today, yeah, we're talking about the obelisk today, and here to help me, because I really needed the help, we have RestaVT. Resta, can you please introduce yourself?
Hello, hello. I am RestaVT, resident ocean explorer investigating the mysteries of the Bermuda Triangle. At least that's my lore.
Let me know if Resta's audio needs any editing. Sorry about that. But yeah, sorry.
Oh, yeah, it's my gain. Okay.
Yeah. Yeah. Please let us know if Resta's audio needs adjusting. Hey, Brady.
Yes. Mic check. 1, 2, 3.
Mic check. 1, 2, 3. Yeah. So Resta's background as a marine explorer is not just a part of her lore. Resta, can you explain what your background in biology is and why I reached out to you in particular for this Obelisk stream?
So, before venturing into the waters of content creation and VTubing, I once upon a time thought I was going to continue in academia, being a bioinformatician, and in particular, marine microbiologist. And so, for one thing, bioinformatics, you can think of that as like computer science plus big data from biology. And it very often overlaps with the field of genomics, which is the study of genomes. And there's lots of variations on that, like transcriptomics for RNA, proteomics for proteins, etc. And I previously did a bioinformatics collab with Phi, with my rebrand as a pillow VTuber way back then. Now I'm trying to actually like lean into my marine science background, and yeah, here I am.
Yeah, that's really exciting. I've turned Rest up quite a bit. Hopefully that has adjusted the gain appropriately. But I just want to re-emphasize that, like, you know, I wanted to do like an Obelisk follow-up because I love the Obelisk so much. You're probably here because you watched that tiny virus video that I did, or, you know, the pathogens that get smaller than viruses, whatever I ended up calling it. But in reality, I was like a tourist. I'm not a geneticist or genomics person at all. I read the papers uncritically, meaning I read the author's words, I just said, okay, and then I put it in my video, right? That's about the extent of research I did, because I did not have the tools or the expertise to actually carefully look at how this research was being done. And so luckily for me, I knew someone who had that sort of background, and I'm very excited to get into how Obelisk research is actually done, which I think is really interesting. Okay, so let's go ahead and get started. The hunt for obelisks, all right? So the paper that we are going to cover is linked in the stream description. It is this paper that I cited for the obelisk segment of my video. Viroid-like obelisk agents are widespread in the ocean and exceed the abundance of RNA viruses in the prokaryotic fraction. I did reference this paper quite a bit in that video, but again, I did so in a way that was pretty uncritical. I just looked at the paper and said, okay, I trust you, and then I moved on. And then I sent this paper to Resta and I went, hey, could we maybe talk about this on stream, because I don't really fully understand everything that's happening here. And then Resta sent me like 2 pages worth of it. I think I was on vacation. I think I woke up to like 50 messages or something from you about your thoughts, and I was like, thank goodness. Because I don't really have all that many thoughts about this paper, but I'm glad that Resta does. So let's go ahead and get into it. So the first thing we have to remind ourselves is what actually is an obelisk. And this picture right here in the middle, this is Toxo's interpretation of an obelisk. I said, draw me a spooky circular rod-shaped RNA, and this is what she came up with. But if we were to actually look at what a real obelisk looks like, it's this thing on the right. And this is one of these obelisks, right? They are rod-shaped, but they're not quite as simple as the cartoon that Toxo had drawn out. And so what actually is this? These are single-stranded circular pieces of RNA. So not DNA, this is RNA. So this is still a molecule that contains genetic information, but not the one that we use to store our genetic information. So these are a bit unique. It forms a rod-like secondary structure. So as you can see in the real picture of the obelisk, it has this like Evangelion angel kind of vibe, right? This like symmetrical, spooky horror kind of shape, in my opinion. And these obelisks are unique because unlike viroids, they actually code for real proteins. Although I don't think they've actually crystallized the protein yet, but they do know that there are sequences in this RNA that could potentially code for protein, and these proteins were given the family name Oblins. 1,000 nucleotides long, exactly. Like, they're pretty significant. They have no existing phylogenetic relationship with any other known life, and they probably didn't come from a, that basically means they probably didn't come from viruses, right? So Resta, did this kind of surprise you to learn? Well, okay, let me back up. Resta, how much did you know about obelisks before venturing here with me on this journey?
I was aware that there are a bunch of strange RNA viroid type things out there, but I think this might have been the first time I heard of Obelisk specifically.
Yeah, they're not really that prevalent in modern biology. They're more of a curiosity or a fun fact because the foundational paper that discovered them only came out last year. And it kind of surprised me that there's anything out there that doesn't have a significant connection to anything else that exists on planet Earth. So that's what really drew me in to how unique and strange these things are. Okay, so I have a confession to make. Right, I already did this confession, but here's a slide. This is a paper I skimmed for my last video, right? Just because I didn't have the background, I'm not a bioinformatician. How do you say that word, Resta?
Bioinformatician.
Bioinformatician, thank you. So I took the authors for their word, and wanting to know more, I recruited our resident deep sea explorer, Resta. Okay, so that's why we're here tackling this paper together. It just popped into existence one day, I guess. Yeah, I mean, as far as we know, right? So I just want to emphasize that when I read for videos, it's different for reading from streams. I really love doing both because when I read a paper for a video, I get to just relax. I get to drink a cup of tea. I get to just enjoy the paper. But then for streams, I'm really tackling the paper. I'm really trying to understand it at a very different level. It's a very different experience, and they both are rewarding in their own way. Okay, so we're going to talk a little bit about how obelisks were discovered. And this is something that I didn't really go into the video. So if you're here looking for more obelisk stuff, here it is, right? So I'm going to butcher this name. I think it's Jaludev et al. Last year. They were interested in mining the human microbiome for viroid-like elements. So obelisks are a relatively new discovery, but viroids are a thing in biology, and they have been a thing in biology for many, many, many years. So viroids were getting a lot of press for their presence in plants, but there weren't a lot of these viroids in mammals, which Jaludev et al. Found to be interesting. And so they asked this question: what if we're missing the mammalian viroids because mammalian viroids are evolutionarily unrelated to plant viroids, right? This is their hypothesis. Maybe we can't find them in mammals because the mammalian viroid might look very different than the plant viroid. So they made their tool. They called it VNom, although to me it looks like VNom. And I think, Resta, you made a really funny joke in our chat. Do you want to say what your joke was?
VNom could totally be a VTuber group name.
Yeah, VNom could be a VTuber group. I did find that very funny. This is apparently short for Viroid Nominator. It's some kind of algorithmic tool that allows them to detect the presence of obelisks, which I didn't really understand too much on my own. But we're not going to get too deep into VNom, unless Resta, you have extra things to add. But for me, I only understand this as like a way to pass molecules through this program to find obelisks. But I do want to take a look at their general approach here, right? So their hypothesis is that the mammalian viroid looks nothing like the plant viroid. Okay, that's their hypothesis. So how do you find something that is unrelated to anything else that you've ever seen? So this is, if you've never been if you've never been a viewer of my streams, this is something that I like to do, is to engage the audience at a deeper level and tell you to be the scientist. That's what this YBTS marker is for. So VNom is a program that these people compiled that allows them to search for viroid-like objects. And the thing about their hypothesis is that this viroid does not look similar to the plant viroid, they think. So you can't just say, okay, find me everything that looks like the plant viroid, because their hypothesis is that it doesn't look like the plant viroid. So what are some general properties of viroids? What are some general properties of viroids, chat? This is your pop quiz. I know you're here for my Viroid obelisk video, so you've definitely watched the video. How would you describe a viroid? What properties could you exploit that a viroid has that you can use a program to algorithmically search through and identify potential viroids? What are some properties? I'm wondering if chat can get it. Replication. Yeah, viroids can replicate with the help of a host. Small, certainly small. I'm sure length requirements are to be kept in mind. Circular, made of RNA, no protein coating, definitely all true. Very good, very good. Yeah. So we're remembering the properties that viroids have, and there's something that chat has said so far that the scientists can exploit. They have an exploit that they can take advantage of to find these plant viroid-like things. So it's good that you have that there. So the property that these scientists are going to be exploiting is the fact that they are circular. And this ends up being really, really important. Yeah, Resta, are you raising your hand?
No, I was just getting some water.
Okay. Okay. So these authors are going to exploit the fact that viroids are circular. Okay, they're circular and they're RNA. And I want to sit with that idea because something that I like to do on my streams is to basically help you walk in the shoes of the scientists because it makes their discoveries even more amazing. So I'm not going to tell you the answer right away. I want us to pause together and just appreciate how are the researchers going to exploit the fact that viroids are circular to be able to write a program to identify viroid-likes. It's interesting. Do viroids have protein or is it only the RNA part? They're only RNA. Is it like a plasmid so they can test them like a plasmid? Well, it depends on what you mean by that. Okay? Yeah, it's interesting, right? So they're going to exploit the fact that they're circular to build their program. And how are they going to do that? We will go into that here. Okay? Bacterial genome is also circular, isn't it? Is it different aside from protein coding? So the bacterial genome is actually chromosomal. Like they're well, let me not get this mixed up. I'm not a bacteria expert. Resta, is the bacterial genome also circular or is it just plasmids? Do you happen to know?
There definitely are bacteria with circular genomes. I think for the bioinformatics collab, we were looking for OEC. If you might recall.
Yeah, OEC might be a plasmid though, or it might have been on a plasmid. So I'll say that I'm not entirely sure that all bacteria have circular genomes, but I'm sure that is the case, or at least some of them. A computer program can easily find circular DNA/RNA by looking for overlapping 5' and 3' ends. Ooh, interesting. Okay. Interesting. Very interesting. Okay, so what might that look like? So how do you identify circularity? Okay, here we have a circle. How would you describe a circle, right? How would you describe a circle, chat? I know Resta knows how to describe a circle, but how would you describe a circle? Think about what it means to have a circular piece of RNA. And for those of you who are comparing this to plasmids, I absolutely agree that techniques used to identify plasmids can be used to identify these circles as well. They rely on a similar principle, and that has to do with the concept of circularity. Behold the circle, exactly. It has no beginning and no end. Okay, it's closed. Okay, it has no beginning and no end, which means what? Periodic. Okay, we're getting somewhere. It's periodic. It has no beginning and no end. So what if you had to read a book that was in the shape of a circle? How would you know that a book that you are reading is circular? Thank you for the circle figure. Dismiss some of my confusions. That's what I live to do. I'd iterate through the chain and see if that elements keep repeating in a loop. It's a circle. Oh, interesting idea, right? If you had to read a circle as if it were a book, you would look for something that repeats, right, in some sort of periodicity. And so the authors are going to take advantage of that kind of idea, the fact that there is no beginning and no end, right? So imagine a purified sample of RNA isolated from the gut microbiome. This is what the original authors were doing. So we're not in the ocean yet. To analyze this RNA, it is typically amplified and conserved by converting it into DNA. This is not really that important, but it's just a general thing that happens when you're trying to analyze RNA. And then how do you identify circularity? And I think, chat, you did a very good job at describing circles, which is not arbitrary for this problem. The authors are going to take advantage of the fact that there is no beginning and no end to a circle. So imagine you have a circular piece of DNA or RNA. This is DNA. I realized I used T's. If I were to be more accurate to it being RNA, replace all of these T's with U's. This is me making the slides like 2 hours ago. If you were to read this sequence in a linear fashion, and I were to start at 12 o'clock here, we would get AGCTAGTTAACG, right? That's what this sequence is, right? This is the linear read of this strand of DNA. So if this strand of DNA were a line, and you were to start reading from this point, you would get, and you were to read the first 5 characters, you would get a certain contig. You would get a certain sequence. The first 5 letters of this book are A, C, G, T, and A. And if you begin reading at a different part of the book, then you would see something else, T, T, A, A, C. But what if you had an actual circle? If you have sequences that have a perfect match between the start and the end of a sequence, it implies circularity, right? It implies circularity. So this is the first step. There are probably others in the paper, but I'm kind of too dumb to figure them out, and I think this is probably the coolest part anyway. So the authors used VNom to plumb the depths of the Integrative Human Microbiome project. They have this program that allows them to check for whether or not there is a beginning and an end. And so if you have something that's circular, notice this read. So if you begin reading here, ACGAG, this read cannot appear in this linear sequence because lines have a beginning and an end. ACGAG does not exist in the linear read, only in the circular read. Does that make sense, chat? Does that make sense? Hey Z, welcome to the stream. Z, another member of V Theorem, our group of VTubers that does science communication content, also a guest on my video about giant viruses. So, so you see how this kind of difference, the fact that a line has a beginning and an end, but a circle doesn't, allows you to see that if you start reading from different positions, you end up getting different reads that are impossible for a linear sequence to have. Yeah. But if you try to find a circular pattern, how do you know it's not a big circle with many repeating items or just a super long open chain? And that's an excellent question. I don't actually know the answer to that, but Resta, do you have a response to that?
Well, if you have many repeats, that tends to make the assembly, which you may visualize as something called a De Bruijn graph, like very messy, because essentially, when you have repeats, you get this very strange loopy structure as a product of the algorithm that you use. I think we have a slide on that.
Yeah, we do have a slide on that.
Something?
We do have a slide on that. Yeah, so there's a comment saying, depending on where you start to read, it could still be linear. Well, assume that you have this as the canonical sequence. If there's a beginning and an end, you can't find this particular subsequence, this ACGAG. Like, it doesn't matter where you start in this line, you cannot find this particular arrangement of letters. That's the basic idea. I'm not going to pretend like I'm an expert on this particular thing, but we will look at the De Bruijn graph in just a little bit to maybe see, and I'll be learning alongside you because I don't fully know myself. It's an excellent question. It's an excellent question. Okay, here it is. Okay, by the way, I think I mentioned it to Resta before, but I dropped bioinformatics when I was in college. It was too hard for me. So I greatly respect Resta, and I'm very happy that she's here with me because I would not be able to do this stream alone. So how do we know that things are a circle? Basic answer for a dummy like me is because computers. Because you can assemble something called the De Bruijn map. And Resta, do you want to kind of walk us through what this figure is?
Sure. So this figure is from the VNom paper, yeah? And so in their search for obelisks, the authors of the Ocean Obelisk paper use this program to find.
Sorry, Resta. It's not. We're not talking about VNom quite yet. We're only talking about the overlap matching in the Bruijn graph.
Gotcha. In that case, when you sequence DNA or RNA, you end up with a whole bunch of short sequences called reads. Those reads themselves will often overlap, and you'll have many duplicates of reads because you'll sequence the same sequence multiple times. That you'll refer to that as read depth. And then from those reads, you'll assemble larger pieces of the puzzle called contigs. You can think of this process as like when you have a jigsaw puzzle. The individual pieces are reads, but then you kind of end up with these big chunks after a while. Those are kind of like contigs. Then from there, if you want to assemble things further, you'll often assemble what's called a de Bruijn graph, where the nodes in the graph are k-mers, that is sequences of some specified length k, and they will overlap by some amount and produce a graphical structure that can be neat or it can be messy. It depends on what context you have. Sometimes you'll also find that the structure becomes circular, or other times some DNA will be so fragmented that you'll only have like a short linear piece.
So if I were to try to take what you're telling me about how this process happens and kind of put it in my own words, through enough, if you have a sequence of enough length, it would be unlikely for there to be a mistake in stitching together these lengths, right? Because if you see in this graph where they're showing the assembly process, so say, for example, you have these short reads and then you get blocks, chunks that are length 2. And as Resta told me before stream started, it's kind of ridiculous. This is a purely for an example, right? You wouldn't normally have reads that are much longer than 2, but if you have a length of 2, this helps you understand it conceptually. So say you have a bunch of reads, AT, TG, GC, AG, CA, CG, and you try to stitch them together in a way that makes sense. One way that it can make sense is through this path finding, where you are stitching overlaps in a way that produces one sequence, and then you get a contig. And if you have a complex enough set of reads, there are only so many valid ways you can stitch together that sequence. Am I getting that correct?
In essence, yeah. And of course, in actual assemblies, you will vary that k-mer length. There's do I remember? But there's a specific method for looking at the distribution of, I believe, read lengths. But from that graph, then you determine what would be an appropriate k-mer size for your de Bruijn graph.
I have a question, Resta. Is this process perfect? Like, do you get the correct answer, or do you get probable answers? I think that's an interesting question.
You get probable answers.
Oh, okay, okay.
Unless the starting reads are in a certain way. Like you can make a de Bruijn graph that has that perfectly represents the original genome if you cut it up a certain way, but that would not happen in real life. I'm just saying it's mathematically possible.
Okay. So I think that might answer some of chat's questions about how does this definitely prove it's linear or definitely prove it's circular. So to answer that, I think, and you can correct me if I'm wrong, you're more limiting the chance. You're isolating what is the most likely confirmation of this garbage that you get. You try to stitch it together and you find that it is overwhelmingly likely to be circular or more likely to be linear, but it's not a perfect thing every time. Okay.
I feel like there's probably a bit more that was done to assess circularity, perhaps. Who knows? Because just relying on the de Bruijn graph alone might not be sufficient for a published paper, perhaps.
Yeah, I did mention that earlier, that there's other stuff, but then my brain started to check out. Like this is the basics here. In terms of my there's a comment in chat asking about my background. I'm a neurobiologist who does immunology for fun, so that's kind of where I'm at. Okay, so what did that foundational paper find? They found that when you try... Okay, so let me back up just for people who might have tuned in. The original paper to have discovered obelisks, they were trying to see whether or not there were viroids in mammals, right? Because viroids have been known plant pathogens since forever, but no one's really seen a viroid in a mammal. And the reason why they think this is so is because the mammalian viroid might look very different to the plant viroid. And if it does look very different to the plant viroid, you cannot rely on genetic similarity to be able to find the mammalian viroid. Instead, you have to do some clever things trying to find circles, essentially, within the metagenome of the Human Microbiome Project. So, before we said, tumors, that's not tumors and cancer, but 2-MER. That's not really practical for contig assembly. So instead, they're using 40 tumor matching, right? So they're checking for much larger chunks that line up with the idea of circularity for these obelisks and other data sets as well. So, they were able to find obelisks not only within the human microbiome from the publicly available data, but they also found the obelisks in medical samples, right? So human stool, they found obelisks in. They also found one in a fox autopsy, which I found to be pretty interesting. Not all of these projects had obelisks, so they're not quite ubiquitous, but they are abundant. They I said for YouTube that they're everywhere, but they're not really everywhere, but they are more common than you would imagine. More common. So if there's no overlap, it's linear, but if there are signs that it's overlapping, it's circular RNA. If there are signs it's overlapping, then it might be circular, probably is circular due to math reasons. Okay. So this is kind of fun. You can read the paper and you can actually find the authors jumping for joy. It's so cute when you read a paper that's written in this very academic language, but you can feel the author's hyper excitement when they were looking at these obelisks, because not only did they find the obelisks and all this publicly available data, they found obelisks collected from samples at their own university. So they scrambled to collect microbiome data from their own academic community, right? They were like, hey, the data that this lab here on campus generated, we found obelisks there. Why don't we just start swabbing people's mouths and stomachs and see if we can find obelisks that are lurking amongst us right here at this university? I found that was so... that's so nerdy. That's so nerdy and so funny. And as they were probing these other samples, they found obelisk beta. Like, if we weren't going into Chunibyo language enough, this other obelisk called obelisk beta, which is an obelisk-like entity with a low homology to obelisk alpha. And you can see this is why people have failed to find the mammalian viroid before. Like, obelisk beta is only 41% similar to obelisk alpha. It's a little simplified. And the overlap in sequence is even lower for the protein sequences of Oblin 1 and Oblin 2, right? Like, the reason why these are so hard to find is you cannot rely on just comparison. You have to rely on some different tricks. And I think that's really, really cool. All right. So they were very excited to find the mammalian obelisk, but they weren't entirely sure where in the mammalian body the obelisk was hanging out. They know that they can find it in the microbiome, that when they collect samples from the stomach, from the mouth, etc., that they can find the signature telltale signs of obelisks existing there. But not anything more than that. Are they found in host cells, bacteria, fungi, archaea? These are all residents of the human microbiome. They could also be free living, but that's really, really unlikely because existing hurts RNA. I make this joke every time about RNA, but if someone sneezes in the lab across the room, all of your RNA will degrade. RNA is fantastically easy to destroy. It's amazing that we even exist today, given how fragile RNA is. So the original authors wanted to find where these obelisks were hanging out, and they found them in one particular host, Streptococcus sanguinis, this chain cocci bacteria. But weirdly enough, the obelisk that they were able to isolate for sure only coded for one of the two Oblin proteins, only Oblin 1, but not Oblin 2. So there are obelisks out there whose location we don't fully understand, who are supposedly making these two proteins, and we can't find them. So weird, so weird. Bizarre, bizarre. New hazing ritual, swab your cohi for their obelisk, true. Contribute to the greater good for biology. Very, very interesting. Resta, did this strike you as unusual? Because you are more of an ocean person. Does any of this sound familiar to you, or are you surprised by any of this so far?
Well, sampling like in your local area, actually, it's common within metagenomics. I hope that's what you're asking. Like, for example, there are studies about the urban microbiome where people will hire volunteers and friends with swab and sample kits, and they'll go out into their cities, like swabbing, like benches and cements and random poles, especially things that people touch a lot, like ATMs. So hearing that the authors scrambled for data within their university, it's like, oh yeah,
that makes
that's
sense.
common.
That's super cool. I love that. 7cube asks, is DNA notably better for stability? The answer is yes. You can leave DNA as long as it's purified and is free from enzymes. You can leave a sample of DNA on the bench at room temperature for a pretty long time before you start seeing any degradation. I'm talking about like days to weeks. Like DNA is pretty stable as long as it's pure. Okay. The obelisk is still full of mystery. We don't know what they do, and we don't know where they are. we can limit the possibilities of inspecting where to look, right? Like we know they're in the microbiome, but we only know one bacteria that houses them, and it's not even all of them, right? We know that there are obelisks that code for two proteins, and we can only really isolate an obelisk that only codes for one. So these guys, and there was a YouTube comment that I think I really have to point out that said, do obelisks even count as pathogens? And that's an excellent question. I mean, probably not. If we can't identify a disease that these obelisks cause, are they really, really, really pathogens? Maybe not. Maybe they are. Maybe they slightly decrease fitness because you have to spend cellular resources to even make them. Maybe they're beneficial. I don't really know. No one really knows. These are really wild. And I love how mysterious they are. And I'm happy to be in an era of biology where hopefully in a couple of years, somebody will figure this out. So what do obelisks do? We know we don't know their full purpose or impact on the biosphere. And so this, I think, is the biggest mystery of all. And again, it's unknown whether or not obelisks even cause appreciable disease, right? Oblin 1 seems to be able to bind RNA, and I don't really know why. One hypothesis is that you need to do this for replication. And, you know, if we look at the other pathogens, quote unquote, not pathogens, quote unquote, the actual pathogens I mentioned in that video, some of those RNAs have ribozymal function, that it is a piece of RNA that itself can catalyze a chemical reaction, notably cutting itself into pieces to, you know, when you make a batch of sweet rolls or something, you roll out your dough real long and you cut equal segments so that you get distinct bonds. It's kind of that way with certain molecules of RNA. So we know Oblin 1 seems to bind RNA. I think I've read a paper even showing that they do have ribozymal function, but I don't have that citation with me right now. Oblin 2 seems to act as a leucine zipper adapter. So by adapter, this is not like a specialty science word. I literally mean an adapter. So a protein that helps other proteins bind to each other. And of course, these are both putative. That's a fancy vocabulary word for you today. Putative. That is just a hypothesized function, not confirmed yet. So scientists, based on the structure of these hypothetical proteins, again, hypothetical structure of these hypothetical proteins, are making guesses as to what these proteins actually do. Yeah. So neither protein seems to have an obvious pathological mechanism, right? These proteins, because they don't look like anything that causes disease, because they don't look like anything else, it's really hard to say what these actually do. All right. So here we get to introduce Resta's like actual real specialty here, is combining the fact that we are looking for these cryptic things that can only be detected by using genomic methods in the actual ocean. Like, I could not have asked for a better partner today discussing the obelisk here, because again, I breezed through this paper. Okay, are they functional?
Appreciate the invite.
Yeah, thank you for coming on. So cool. Okay, so the paper, I immediately was confused and kind of annoyed at this paper because this paper doesn't explain anything, which really makes me sad. But thankfully, Resta actually has experience in this and can walk us through how these samples were actually collected. Now, I do know that they mentioned the use of a tool called the CTD probe, which stands for Conductivity, Temperature, and Depth Probe. So you go on a little boat and you drop this thing in the ocean. And so that is the general way that these samples are collected. And Resta, do you have anything to add about this particular methodology?
Yep. CTD rosettes often actually do other things aside from measuring conductivity, temperature, and depth. One thing, for example, is they'll actually measure chlorophyll levels by using some kind of light-based method, like shine lights. You have chlorophyll concentration. It refracts. Sorry, I'm not a physicist.
I actually know this one. So the way that they detect chlorophyll, and so let's maybe back up and ask. So why are the researchers interested in chlorophyll? What does chlorophyll tell us about the ocean? This is a question for anybody to ask, or like, what is chlorophyll an indication of, audience? Hold on, a little boat? That's a cargo ship. That is true. This picture is like this, but I think there are other boats that are maybe not as impressive that do this kind of stuff too. Yeah. What does chlorophyll in the ocean? Why do we care about chlorophyll in the ocean? Algae concentration, oxygen levels, cyanobacteria, presence of light. Interesting. Yeah. So if we back up and we think, okay, why do we need a CTD probe to begin with? Why don't we just take a little cup and just start scooping ocean water and just analyze that? A CTD probe allows us to actually determine which layer of the ocean we're actually sampling. And as Resta said, one of the layers that we're interested in is this layer with chlorophyll in it. And so chat is right on the money with cyanobacteria, oxygen, photosynthetic life, etc., etc. So we are wanting to know, if we're trying to find if obelisks exist in the ocean, we have to do it in a smart way, right? We can't just take a little spoon and start scooping up from the top. We want to be able to sample different layers. And chat, you're completely right. We're sampling the photosynthetic layer, which is indicative of the fact that there are photosynthetic organisms living in that layer. As you said, algae. Okay, I want to back up because I don't know anything about the ocean. I'm assuming that means algae, cyanobacteria, etc. Phytoplankton. Yeah.
Yeah, phytoplankton. Yeah, definitely. Because those organisms are essentially like the base of your ecosystem. So by measuring chlorophyll, you can kind of use that as a proxy for primary productivity.
Yeah.
Another interesting idea here is that in the water column, you have all these different layers. Now, like you might be like layers, it's just a bunch of water, right? But in the ocean, you'll have different like kind of transition zones. So if you think of like, let's say Minecraft, you have like these different biomes where there's like the desert and like the mushroom forest and whatnot. But in the ocean, you have, for example, the deep chlorophyll maximum, which is where if you measure chlorophyll while lowering down a CTD rosette, you'll see a peak in chlorophyll there. If you measure temperature, you'll also see a sharp temperature change at what's called a thermocline. There's also something called a halocline, where you have different salinity changes. and along the halicline, it changes very rapidly. And this creates very interesting ecological zones where different life might be observed in one place versus another place.
Yeah, and we'll actually have a look at those layers further on into the slide. But now here comes to the point where the paper drops this thing where they say a lot of the data that comes out of this mining of like sampling of these different layers comes from something called the Terra expedition, which is something that Resta geeked out a lot about in our private chat. But I don't actually know anything about the Terra expedition. So I'm going to hand it over to you. This is a picture of the boat that I found. It says Terra on it. And I'm just going to let you go, Resta, because I don't know anything about this expedition. So enlighten us as to what this accomplished.
So, the Terra Ocean expedition is kind of famous within the like marine ecology sphere and also the bioinformatics sphere a bit, because they use this modified sailboat and traveled the globe looking at so many different ecosystems from the tropics to the Arctic, and they sampled water like everywhere you might imagine. And so just seeing that data, which itself is kind of famous, being also used in the study looking for obelisks, like, yeah. Like, yet again, like the Terra Ocean data is becoming invaluable to the scientific community.
And someone had mentioned that the cargo ship looked huge, but I don't know, to me this boat looks pretty small. I'm actually surprised that such a cornerstone of ocean microbiology was built on a ship that looks, this ship doesn't look anything that special, and yet it has been, from what I'm hearing, a very, very important expedition for the sake of ocean microbiology. Is that correct?
Certainly. Also, in addition to those large CTD rosette rigs, you'll also sometimes sample water with smaller setups that use peristaltic pumps. And I'm not sure if Terra used it, but I feel like they might have, because obviously there's no super big crane on this boat, but I should look into that sometime.
Very cool. Very cool. All right. So we're sampling the ocean, and here is what I took from the paper, that they were sampling these specific layers, and I don't really have anything to add to these. But there's a surface layer, which is very short. I think it goes up to like 7 meters down. And then you have the epipelagic mixed layer. Don't know what that means. Deep chlorophyll maximum. This is something that they wanted to pay a lot of attention to due to the diversity of microbial life in here. And then the mesopelagic layer. And I don't know what any of these mean. So Resta, do you have any additional insight as to what it is these layers actually are? If, for whatever's relevant to the story about obelisks.
Well, really, what was highlighted in this particular paper was a deep chlorophyll maximum. I guess I'll spoil it a bit, but they were, the authors were curious to see if we'd find more obelisks within that particular layer, because then that could provide a clue as to what, where the obelisks are, what they're doing. The authors didn't find that, but what they did find is that obelisks were kind of ubiquitous to an extent, at the many different depths. They'd find obelisks, or rather predicted obelisks in the data.
Now I'm wondering if I have my slide wrong. Hold on, maybe I made a mistake. I thought they did find an overrepresentation of deep chlorophyll maximum, but it's not.
I thought they didn't.
Hold on, maybe we're. Give me a second. I would like to pull the paper up. Hold on.
Let's go for it.
Yeah, give me a second while because I think there is a figure. Well, we'll continue because, yeah, as you said, that's a bit more of a peek later into the paper, and that might not be so relevant right now. So we'll get into that, I think. Okay. Okay. So you've sampled the different layers. What do you do with them? The paper is woefully short on methods on how to convert a water sample into meaningful data. So I'm super sad about this because I don't know how this is done. They collect water and then I'm left to my own imagination. Although I can imagine how they did it, I don't know for a fact. So I'm guessing it's some method of RNA extraction where you take that ocean water, you expose it to mild detergents to break open the cell walls and pull the RNA out. And then probably some method of converting that RNA into DNA to stabilize it, because DNA, it can sit in a fridge forever. Basically, RNA, you look at it too hard and it destroys itself. And then you put it into a fancy machine where you can read whatever that sequence is. And then, that method of analysis, trying to find obelisks, you could use VNom. Again, VNom allows you to try and find circular pieces of RNA, and that's how you can find an obelisk. Okay, but for some reason, and this is really bizarre to me, and I think it was bizarre to Resta too, the authors tried to use VNom on these samples, but they couldn't find any obelisks using VNom. And that was weird. And they didn't really say anything about why they couldn't get VNom to work. So they pivoted to something else. Yeah. If I was the inspiration for drawing the obelisk so damn creepy, I think of obelisks, I can just nerd out here for a second. The whole inspiration behind the Tiny Viruses video is the fact that, like, life finds a way. I don't know if you've heard that expression before, but life certainly finds a way. And I think, there are people who find that very beautiful, and there are people like me who find that a little horrifying. The nanosecond, I said this in the video, but the nanosecond you give a material the ability to self-replicate and mutate, it automatically becomes lifelike. And so the creepiness, I think, comes from the fact that these are ostensibly just chemicals, molecules, that somehow act in a very similar way to life as we know it. This concept of, what is it? Not the Erie Canyon. What is it? What is that phenomenon called? Where something is too human-like and it gets creepy? The Uncanny Valley.
Uncanny Valley.
Yeah. Yeah, the obelisk kind of lives in the Uncanny Valley. So I told Toxo to draw the obelisk like this creepy thing because that's the feeling it evokes in me. Yeah, I hope that explains that. Okay. Yeah. So they weren't able to get VNom to work, which was kind of weird. So they made their own thing, or they used their own thing called Pebble Scout. So I don't know a lot about Pebble Scout, and I didn't bother to do any research into how Pebble Scout works. I didn't have the time. I was on vacation, and I got back last night. But essentially, the too long didn't read for Pebble Scout is that it is very, 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 very fancy Control F. So you can Control F using the original Obelisk database generated by the Pioneer paper, and then Control F over petabase scales. So these are databases containing more than 10 to the 15 bases, which is a number I can't even imagine what 10 to the 15 is numerically, right? That is, if you've done any, and Resta, I've assumed you've done search algorithms at some point, maybe?
Yep.
And
I could... Oh,
yeah, what's up?
I could add a bit here, if you like.
Please, go ahead.
I really like how you described it as a fancy Control F, because in the field of bioinformatics, more and more sequence data continually gets added to databases like the SRA Sequence Read Archive. And so actually searching that database poses quite a problem. And Pebble Scout was developed by some people, probably under some federal grant, to help scientists look through the ever-growing amount of sequence data on that website. And since from a previous paper, the known obelisks sequences were published, the authors of this ocean obelisk paper just had to take those sequences, put it in the site, and see what pops out.
Yeah. And again, 10 to the 15, man. that's so much. That's crazy. And to have it be done in a human lifetime scale, I can't imagine, man. Search algorithms are notoriously difficult to write because searching can be quite inefficient, but the fact that you can search 10 to the 15 bases multiple times over the course of, you know, one paper is amazing. It's, you know, I can appreciate how good that is.
Thank you, hash tables.
Yeah. I don't know what you mean, but I'm sure somebody does. I do miss it though. It was fun before I dropped bioinformatics. The idea of like big O, like, right, that's the thing. Like how your algorithm scales with complexity, and search algorithms have to be very, very fast, because if you don't scale them up to huge databases, they're going to be very, very slow the larger those databases get.
For sure.
Okay, so they use their Pebble Scout program, and they use a bunch of other programs to actually visualize what these obelisks look like. And you can kind of see that obelisks, not only do you find them in all the oceans, right? This is Pacific Ocean, this one is the Atlantic. They found them in every ocean they looked at. But they also look different to each other, right? This obelisk from the Arabian Sea, this has like two forked ends, it looks like. It looks like this Arctic Ocean one has a little hat and a little like skirt in the middle here. And then this one, I think this bottom one is from the Pacific Ocean, and this one is really, really tight on forked patterns. Like these look really, really different to each other. And as you know, as I pointed out in the video, and as many commenters have pointed out, these shapes remind me of like Evangelion or biblical angels, right? There's something uncanny about the way that these look. They kind of freak me out. Yeah. But not only do the obelisks look different, they also make proteins, or again, hypothetical proteins, because no one's actually purified this protein yet, but their hypothetical structure looks different than the oblins that were initially discovered in that previous paper. So in every insert box here, we have an obelisk from the original human microbiome dataset, and on the right we have the ocean version. So if you look at Oblin 1, this is one of the Oblin 1s found in the human microbiome. This is very different than the Oblin 1 found in the ocean. Similarly here, one version of Oblin 1, they all look really different. Like the variety here is insane. Like these structures look very, very dissimilar to my eye, at least, as someone who isn't like a super crystallography expert. But like if you're talking about, you know, is my human MHC the same as a mouse MHC? If you overlay those two proteins, you're going to find that they look extremely similar. And you can already tell, you don't need to overlay them. These are quite different. These are quite different. Very, very bizarre. Very bizarre.
I remember in the original obelisk paper, the real important kind of structure is where you have like those three alpha helixes almost like in a triangle, and then a fourth alpha helix that's like wedged in there. Because I remember the rest of the structure in that paper, like they're saying like, oh, we can't even like fully get a good prediction of its structure from AlphaFold. But what they did get a good prediction for was that particular, was the alpha helix structure at the bottom. Cool.
And that makes sense. Okay, so if you've never seen a crystal structure before, let me walk you through what we're looking at here. And we'll actually play with this structure in just a little bit, because Resta has so kindly prepared a little interactive thing that you can all take a part of, which is very, very exciting. But again, if you've never seen a crystal structure before, I mean, I've showed them in my videos before, so you've at least seen one of them probably. But just to walk you through, here, in this region, you have what looks like a spaghetti noodle. This is an area that the folding program has determined is essentially non-structured. If you imagine this protein floating in water or a saline solution or something, this thing is going to be very, very floppy. It's going to be like the floppy guy that sells you tires. What are they? A wacky inflatable tube man, right? But down here, when you see these thicker helices, this is an indication of a region of the protein that is ordered, as in this protein part does not move all that much. It is solid in a kind of way. So you can see that the folding structure, the predicted folds, show that there is this common 4-helix bundle down here in Oblin 1. And yeah, they might all look kind of different, but they share a similar superstructure, I guess you could call it, where it's 3 helix bundles and then a 4th helix kind of tucked in somewhere in these 3 helices, but the rest of it looks very, very different. Spaghetti noodles that flip and flop all over the place, which is very, very interesting. Okay, so The authors used something called CollabFold to predict the Oblin structure. Again, nobody has purified. We don't even know if the obelisks actually make these proteins. Like, they have the gene for it, but has anybody seen the protein? No, not yet. This paper came out in 2024, so maybe somebody's trying to, purify or make the Oblin protein. But for now, what we have is predictions. And this is where the use of AI and machine learning and all these high-powered conceptual models can actually be a boon to humanity instead of ruining it. CollabFold is very, very cool. So let's play with it now. Let's see. Resta sent me a link, and I will pin the link in the chat. And you can open up this notebook and play with the protein for yourself. So let me go ahead and get that link for you all, and then I'll play with it on screen as well. So here is the collab notebook. I'm going to pin it, and I am then going to pull it up on the screen. Hopefully everything is good and fine. There we go. Okay, so let's walk through what it is we have here. And let me go ahead and hide that PowerPoint so we can all look at the same thing together. And here we have the start of the notebook. This is AlphaFold. This is the Nobel Prize winner of 2024. I think AlphaFold won last year. Can correct me if I'm wrong. But essentially, this is a super high-powered AI tool that allows us to predict the intricate ways in which a protein can fold. And you can see, thanks to Resta preparing this notebook, that you can enter whatever protein sequence you want. If you've never seen a protein sequence before, this is what it looks like. You have 20 essential amino acids that get stitched in various combinations to make your proteins, and every single letter here is representative of one protein. It's been a while since I've had all of these memorized, but for example, this particular oblin, the sequence here is methionine. I mean, it's a start codon, so it always starts with methionine. Serine. This is one of the, I want to say, I'm showing my whole, I want to say this is glutamine. I'm going to get roasted by anybody who's recently taken biochemistry. T3anine, maybe? Glutamate, I think is E. Serine, leucine, serine, alanine, serine, arginine, etc., etc., etc. You get the point. Even if I didn't do that completely correctly, you kind of get what this sequence represents, right, chat? Kind of get it. All right. Okay, so you follow the code and everything installs and it does some things of which I don't really know. Is there anything here, Resta, that you want to point out in terms of the settings that are interesting to know? Because to me, it's just input the sequence and then let the program do its thing. But I don't actually know if there's anything else that's interesting that you want to point out here.
Well, in general, in bioinformatics, settings are actually quite important because, like, what we're like, when you do in bioinformatics, you're trying to find something that represents the truth, right? Mm-hmm. But one particular setting, that's just like an on-off switch. Like for like a video game, like you don't really care like what anti-aliasing is being used or whatever, right? But for bioinformatics, that switch could be a very important assumption that could alter the results produced by a particular program. The default settings here suffice for producing Oblin 1 according to the original paper. So we didn't have to mess around with that. But in other scenarios, just as you would make assumptions when doing statistics or more wet lab type research, like having the right settings and flags in bioinformatics is also quite important. Like, for example, if you're building a de Bruijn graph, right? Of course, if you set the k-mer length to 2, you're going to get a crazy mess and be like, oh, this is just junk. But if you set it to an appropriate length, then you'll be like, oh, I'm starting to see some structure of the genome here.
Are there any particular settings in this CollabFold notebook that you want to point out that might be important, or is it just like stuff that you really have to have a good background to understand?
Probably stuff you'd have to go into the documentation for.
Okay, gotcha. Yeah, no problem. So we have here is like I know I'm kind of springing this on you because I didn't mention it before stream, but like, do you know what this graph I'm showing is? Because can you explain what the sequence coverage plot means before I show the actual protein? Because I feel like the audience might be kind of interested as they leaf through this notebook.
Let's see. Sequence identity to query.
Because I'm guessing position is like the length of the protein, like the first amino acid and then the last amino acid on each end. And then sequences, is sequences like the number of reads that they have in that particular part? I'm not going to try anymore.
We didn't input reads. No, we only input the protein sequence.
Gotcha.
I know that essentially CollabFold wraps both AlphaFold and MMSeq too. So maybe at some point they're doing some kind of read stimulation, but as for like what practically this graph would be used for, I don't know.
Okay, no worries. I was a little curious. But ultimately what we get is a predicted structure. So you see some of them here that are kind of neat, and then they're color coded by how confident the model is in assigning those structures. And then eventually we get to a point, if you continue down enough to display 3D structure, here you actually get to play with the thing, right? Let me see if that's actually showing up on OBS. Okay, yeah. Here, if you wanted to, you can actually look at what this Oblin protein, this Oblin 1, might actually look like. Again, no one's actually purified this thing yet, but if you wanted to work a little bit of good AI into your diet, this good AI is showing us with some degree of certainty, at least with this alpha helix bundle down here, what this protein actually for real looks like. And you can zoom in and see the Oblin protein in all of its glory. So the part, I don't know, Resta, if you had read this, because I kind of skimmed over this, but is this alpha helix bundle, is this the part that actually binds RNA? Is this the part that they are hypothesizing has that kind of function?
Oh, I'm not sure.
Yeah, I wish I had read the paper a bit more carefully now that I'm here. But that's something that you could do, beloved audience member. Play with this protein, play with it on CollabFold. And if you have other proteins whose structure you are short protein, let me be very clear before anybody like goes mad with power here. Don't try to do this with long proteins, or you will make the server suffer a lot. This is like a pretty okay length of a protein, but if you want to do a new like here's what I would recommend you don't do. Don't like, and I know if I say don't do it, people will do it, but like please, like from me to you, you know how expensive these things are to like run, right? Servers are not cheap, and they're doing this for free, so be responsible. Okay, chat? Okay, be responsible. And only
Chat,
fold.
put Rubisco in there.
don't put a big thing in there. Don't. Okay, keep it short. Keep it short. It's a request from your beloved AI, your beloved I almost call myself an AI. Your beloved neutrophil YouTuber. Please act responsibly and fold small proteins. They might even like boot you from using the server if you try to fold something that's too big. Yeah. Okay. Collab doesn't allow you to use doesn't Collab allow you to use local GPU? I don't actually know if this site makes you use local GPU. Do you know?
I personally have never used Collab on my own machine, because at that point you might as well just install a Jupyter notebook on your own system, or your university's cluster. Yeah. But yeah, essentially, any programs that you install get installed on computers that Google owns, and it creates a nice environment for your programs. And that's actually one of the big benefits of collab within bioinformatics and kind of scientific communication and also reproducibility, because just installing programs in bioinformatics can be very difficult and often a major turnoff for people delving into computational biology, because it's very easy to fall into something called dependency hell, where like in order to install one program that you need all these other programs, and then if you want to install a second program, you need a whole bunch of programs, but sometimes those programs are the same programs as you need for the first program, and they might have different versions, and things will break quite easily. But if it works in a Google Colab notebook, then you know, okay, it works on Google's computers, and you can share that notebook with other people, which I wish the authors of this paper did, because I tried to actually reconstruct one of the putative obelisks that was found in the Terra Oceans data set, but I wasn't able to do it because the authors did not specify what reading frame nor codon table they used. So
yeah, just another.
I could try to like trial and error that for hours on end, but
that's another frustrating aspect of this paper. As you can tell, there are a couple of frustrations here. Okay, very, very cool. That's awesome. And thank you, Resta, for preparing that collab notebook for us to all enjoy. I thought that was very neat. And as you could tell, I didn't include an Oblin in my own video because I couldn't get it to render in Blender. I don't know, Brady, if you're still here, but I don't know what I'm doing wrong. Brady, who popped in chat earlier, is the creator of Molecular Nodes, which is a plugin for Blender. And he's a member, which I'm very, very grateful for. Like, not only did you release this technology for us to use for free, but and then like giving me five, like joining membership was also very, very sweet. And I get shout out to all the members for helping me make this YouTube thing like my dream job. Yeah, just a random moment of sappiness, but thank you all, like seriously. Okay. So this is the figure where I thought they had argued that they found a lot of obelisks actually in the deep chlorophyll maximum, but maybe that's only for certain seas. Okay. Or certain oceans. I
double-checked the paper, and to quote it, did I lose my place in the paper?
Yeah, I should have the paper up,
yeah, they did find many obelisks within the deep chlorophyll maximum, which is an interesting observation. But then they're trying to look for hosts, I believe, and were unable to find anything on that front.
Trying to look for what now? Sorry.
I think they were looking for hosts. Let me.
Hosts. Yeah. Okay, we're on the same page. We're on the same page. Okay, so let me walk you through what this monster graph actually is. So on the left here, you have a list of all the different obelisk sequences. So every line here is a different obelisk, and here you have the obelisks that were found in the human microbiome, and then here are the ones that were new and found in the ocean. And up here, you have the different samples that are color-coded depending on which layer of the ocean they found them from. So in the gray, you have the deepest layer, the mesopelagic. I don't know if I'm pronouncing that correctly. In blue, you have the epipelagic, which is right under the surface. In yellow, you have the surface. And then in green, you have the deep chlorophyll maximum, sort of where we expect there to be a lot of teeming microbial life, right? And as you can see in the Mediterranean Sea, there is, and the circles correspond to how much of a representation there is relative to the rest of the layers, right? So basically bigger circles means more. So in the Mediterranean Sea, you can see that there are a bunch of obelisk species that are really, really abundant in the Mediterranean and not so abundant elsewhere. This rule doesn't hold true for every obelisk in every sample they find, but I think if you do kind of look at it, you'll see that there are a lot of big circles in the green areas, but I'm not entirely... I think that it is being dragged by a couple of samples having a lot, like down here, these obelisks Like it's so big, it's actually interfering with the other columns, right? Some ocean samples have a lot of certain obelisks, but it's not always that straightforward. Not that straightforward.
Okay. I found the section in the paper, which was, to quote, the highest abundances of obelisks were found at the deep chlorophyll maximum, which led them to believe the hypothesis that obelisks may be associated with phytoplankton, such as cyanobacteria. Okay, that would be a very interesting ecological find. However, CRISPR in silico massive search of viral protospacer match with a database of over 11 million prokaryotic spacers did not result in a successful identification of obelisks and potential hosts.
We are going to get into that point because that is a really big question mark that I find to be pretty questionable. Okay, this is also not the full thing. So there was a comment that said that I feel like there are fewer unique obelisks than I expected. That's because this is a sample. You can download the entire supplementary figure that has the entire list of obelisks, and there are quite a few. There are quite a few. This is just a selection that we can visualize with our human eyes, right? Okay.
One thing when I see this graph too that makes me wonder is like, because in metagenomics you're sampling like all the particular DNA or RNA in a particular environment, that sampling process is very susceptible to contamination. So when I see something like this, I'm like, are obelisks really like everywhere in the globe, or are these artifacts from human samplers?
Oh, we're gonna get into that. Oh, hold on.
Okay.
Hold that thought, Resta. Hold that thought, because that is so interesting, because we came up with the same conclusion, even though we didn't talk about the paper before today. We did a little bit, but like we didn't interpret the paper as much as we are like together. We've reached sort of the similar conclusion, mostly independently. Before that, I'm just gonna say hi to Mylan, who dropped in. Mylan Aikman, fellow member of V-Theorem, psychology and statistics VTuber, part of the same group that I'm in. Welcome, Mylan. And also, for those of you who don't know, deep chlorophyll maximum, we are sampling layers of the ocean for obelisks, and the deep chlorophyll maximum is where we expect there to be a lot of teeming microbial life.
Hey, Mylan. Yeah.
So
sucking up water from the ocean, passing it through a filter of a specific size that you expect microbes to be caught by, and then you put that into little Eppendorf flasks with some kind of like preservative agents. There's probably a better way to say that. And then send them off for sequencing.
But when you look at this figure, your mind heads to contamination as a potential issue. Not saying that their sample is contaminated, right? But it's giving vibes of contaminated. Is that correct?
Yeah, and like with all the fact that they didn't get VNom to work, even though VNom was such an integral part of like defining obelisks for the scientific community, it is it is peculiar.
It is a little thinking emoji, right? It's a little thinkies. That I think this is how you act respectfully to the authors too, because we can't ignore our own skepticism, but the authors might have had a good reason, right? Maybe VNom wasn't working for a very good reason, but because they didn't say what that reason was, we just don't know. So want to be kind to the authors, but at the same time, I don't want to lie to you or myself. Okay, but there is skepticism here, so we'll talk about that. So I think we kind of already talked about the slide, so I'll just blast through it. So yeah, as Chad had mentioned, we kind of expect in the deep chlorophyll maximum there to be things like phytoplankton, cyanobacteria, other diatoms maybe. So
in fact, since the tests, that is the like outer like shell structure, but they're called tests of phytoplankton, which are typically made of either calcium carbonate or silicate, tend to be very resistant to the typical lab reagents you'd use to lyse like normal like bacterial cells. So often in ocean science, we have to use harsher methods, which unfortunately tends to also degrade DNA more.
Which is also an interesting insight because I can imagine that a bacteria that lives in the harsh ocean probably will have invested more evolutionary resources to survive that ocean versus something that hangs out in the warm, cozy 37-degree gut. So here's another You Be the Scientist moment, chat. How would you test for whether or not obelisks are found within cyanobacteria, assuming this data is good, right? This is what I always say when, like, you know, when students start to read papers and they start to realize, oh, I can be critical too. I can point out what they do wrong. That's actually the easy stuff. It's a lot harder to make a judgment call on whether or not the overall message of the paper can be trusted or not. So I want to take a step back and say, even though there are shaky things about the paper, I'm still willing to believe in everything up until the very end, and then I'll reassess my evaluation. So assuming all of the data is not contaminated, how would you test whether or not obelisks are actually inside of these cyanobacteria? It's interesting. This is a tough one. I don't expect many people to know the answer here, although Resta kind of spoiled it. But I think that there's enough jargon in what Resta said that I think most people aren't going to realize what she meant. Gene probes, magnets, certainly an interesting idea. What do you mean by gene probe? Do we name obelisks or just give designations? So they just give them a code name. I think they probably have some automated program that just appends 1, 2, 3, 4, or whatever conditions that the obelisk was found in until further notice. Because sometimes you just give it a traceable name that allows you to easily match that obelisk with the data that you collected. And then if that obelisk ends up being important, maybe you give it a fancy name later. All right, really big magnifying glass and steady hands. Oh, if only it were that simple. Unfortunately, RNA is a bit small to be able to resolve individual bases like that. But imaging, you know, maybe not a bad idea. Imaging of some sort. Although I'm still fishing for an idea that can be translated into the lab here. Fluorescence, aka make the obelisks light up in cyanobacteria. That is interesting as well. But now we have to think about something that Resta has to think a lot about, but I don't have to think a lot about, because when you deal with anything that ends with omics, you have to make sure that whatever strategy you implement is scalable. Remember, we are searching for hundreds and thousands of different possibilities, different obelisks, right? And if you say something like, oh, we'll take these cyanobacteria and we'll use a genetic probe. We'll use a piece of RNA that looks like the obelisk, and because RNA likes binding to RNA, if we have a specific RNA for every obelisk with a fluorescent probe attached to it, if the cyanobacteria lights up, then we got a hit. And I would say that's not a bad idea. There are issues, however. One of the issues that I can see being something that could be considered is the fact that obelisks are rod-shaped. They bind within itself, which means you have to do some tricky things to get it to be single-stranded again, because it's a single-stranded circular RNA genome, that is true, but it is base paired with itself. It looks like double-stranded, even though it's just single-stranded, right? So unfolding the RNA and then getting an RNA probe to stick to it, a little tricky. But also, if you're searching for thousands of obelisks, making thousands of different fluorescent probes, I'm not saying it wouldn't work. I'm saying you have to be a little more clever to be able to do it cheaply. It's not a bad idea. I think it could work, but maybe not the best option here. Good guess. Would you isolate and create your own culture of cyanobacteria and see the evidence of obelisks? That's also an interesting idea. Resta, do you have any insight as to whether or not that's a good idea? Culturing cyanobacteria. Do you have any opinions there?
Ideally, you'd want to observe it like with minimal like interference, like as close to in situ as possible. Because if you start culturing them in the lab, then you know that's a very different environment from actually in the ocean.
Yeah, and I'll add on to that. What if you bring those microbes to a lab and they start replicating? What if the conditions aren't right for obelisk replication? What if you lose the obelisk in the lab, right? So that's also not a bad idea, but there's also issues with that idea because you can't grow every microbe in the lab. There are archaea that can't be grown in the lab, famously, if you watch my archaea video. Yeah, and you might lose out on a lot of variants, right? I'm not saying it won't work. Here's the beautiful thing about experimental biology. A lot of you have ideas that could work in theory. But the beautiful thing about getting a higher education in biology is you start to see which ideas are better than others. So chat, I think your ideas could work, which is great. That means you understand the science well enough to come up with things that could work. And now what I want to help you with is developing those ideas to be better, more practical, more likely to resolve in a hit. So I congratulate you, chat, for coming up with ideas that could potentially work, but I'm going to push you to think a bit deeper because that's not what these authors ended up doing. Although, again, I'm not saying that your approach doesn't work. I'm saying I understand why the authors chose not to do what you guys are suggesting, but I am proud of you for having come up with ideas no matter what. And I really love that aspect of stream, if you couldn't tell. Okay, so let's go ahead and review what property of bacteria the authors took advantage of here. And again, I don't expect anybody to get this, so I am happy that anybody had answers at all. Okay, because this is something that relies on deeper knowledge of biology, which is, not a problem. All right, let me get on my little soapbox. Okay, I don't ask questions for a gotcha moment, right? I'm not here to make you wrong. I'm here to allow you to play in the space of a biologist, because I think you're smart enough to, right? I think that if you are a fan of Phi the Neutrophil, I think you appreciate the opportunity to try. And it is wonderful that you try. That's all I'm saying. Even if you aren't right, your brain got better because you attempted to do high-level biology. And I think that's awesome, and I'm thankful that you are here to join me today. With that being said, what did the authors actually do here? Okay? You could filter by different fractions, use a filter and filter them by size, extract the RNA. But again, as Resta said, you really want as close of an environment as possible to the actual real sample that you collected, so you don't have to lose things along the way or correct things along the way. It doesn't tell you the species as well, right? If you lyse all the cyanobacteria, you all of a sudden don't have the ability to distinguish which obelisk came from which cyanobacteria. The authors can do better by taking advantage of a very specific property of bacteria, and the answer is CRISPR. Oh, the answer is CRISPR. How cool is this, y'all? How many of you remember what CRISPR is? Not the DNA editing tool, CRISPR-Cas9, that we have adapted into biological engineering to bend biology to our will, but the original OG CRISPR that I made a video on. I think it was like my second video ever on this channel, 2 years ago when I started this channel. The third or second video I made was on ancient CRISPR, not our tool CRISPR, but where we took CRISPR from. The answer is CRISPR. Oh, and this is cool, okay? Bacteria have an infection memory, right? They remember being infected by different viruses. How do they have a memory? They store that memory in their own DNA. There's a part in their DNA called I don't remember what CRISPR stands for now. Palindromic repeat something or another, interspersed palindromic repeat, place where they have snippets of viral DNA that gets sandwiched in between their genome, right? So they have a specific part of their chromosome that stores virus DNA. And so in order, and if you break open a whole sample of different ocean microbes, their DNA will remain largely intact. So if you sequence that DNA, you can see which pathogens or which phages, which virus-like things have infected which bacteria. So it doesn't matter that you mixed 10,000 species of different cyanobacteria, because each species of cyanobacteria will have a different CRISPR, system, essentially. You can now trace infection memory to the species. So again, chat, you came up with good ideas. In fact, those are ideas that I think I would have come up with, because I'm not as clever as these authors are, right? These authors realized that infection memory can help them with determining whether or not the obelisk can be found here. And I think That's awesome. Anything to add, Resta?
Just that.
And it's sick. So what did they find? Oh, clustered regularly interspersed short palindromic repeats. Thank you. Yes, that is what CRISPR stands for. But yeah, so that's very slick, right? They can find out which bacteria the obelisks have infected because bacteria will remember a history of infection. What do they find? Nothing. Obelisks did not show up in any infection memory in the samples they tested. And now I'm worried. Let's take a moment to think about why I'm worried. Why does this concern me, chat? I just told you how brilliant these scientists were. They realized that bacteria have an infection memory. They can trace obelisk infection to each individual species of bacteria that they sequence, but didn't find any. And now I'm worried. And now I am spooky. I'm spooked out. This is weird. And Resta also caught that this was weird. Yeah? This is bizarre. Anybody in chat want to tell me or tell me what you're feeling about this result? It's one line in the paper. They don't say another word about the fact that they could not find obelisks in these bacteria. So what might this mean? What could this mean? Yeah, I'm getting a lot of good responses in chat because it implies that obelisks are not infectious and are native to bacteria. Maybe the bacteria don't treat obelisks like an infection. That is true. So I wouldn't say that obelisks aren't infectious because that implies that we know anything about obelisk infection. But I'll say that one hypothesis, which you're absolutely right, Dreamless, and other people in chat, I see you too, you've had this similar response. Maybe obelisks are not processed in the same way as phage are. Maybe the bacteria doesn't treat them like viruses, which also concerns me because that interferes with my idea of what the heck bacteria even do to fight phage. Now I'm questioning whether or not I understand CRISPR because I don't see a way for an obelisk to avoid getting CRISPR'd. So that's interesting, right? So I don't really particularly believe that the obelisk has a way to dodge CRISPR unless maybe Oblin 1, we know it binds RNA. Maybe Oblin 1 protects it from being CRISPR'd? I don't know. Could be. We know that it binds RNA. We know that it might have ribosomal function. Maybe somehow Oblin 1 is doing something to prevent it from being integrated to bacteria. That is one possibility. One possibility. Doesn't mean it's the only possibility, but it is where my brain went to. That is just bizarre, right? Someone else says it could infect eukaryotic organisms. That's true. Maybe we're looking in the wrong species to begin with. Maybe this is real and obelisks just don't care about ocean bacteria. Maybe they prefer to be in a plankton or something. It could be true. It could be true. Let's see. Does CRISPR deal with infectious DNA? It deals with things that don't belong to the bacteria, genetic material that does not belong to the bacteria. Okay, so that's weird, but there are other explanations, right? So why couldn't the authors find all of this within bacteria? I think, Chad, you've done an excellent job listing some hypotheses. These are ones that I came up with, and then Resta, you can add more if you can think of any more. One, maybe obelisks just don't infect marine bacteria. Maybe something went wrong during sample processing, right? This is what Resta was talking about, contamination. Maybe this whole thing is, hinges on the fact that everything was done correctly. And this is the worst one, right? This one would make me the saddest, and it would make the authors the saddest. We don't want this one to be true, right? It would make everybody feel bad. So don't pray for a paper's downfall.
I've actually heard of some papers that came with that had like spurious results because they used reagents that themselves were contaminated, like the lab reagents you buy.
Yeah, you have to be so clean when you deal with RNA and stuff like that, right? Again, no malice to the authors. They probably did the best they could, but it is a possibility, okay? Maybe their earlier samples were contaminated and there aren't obelisks in the ocean to begin with, right? Again, it's a maybe. I hope everything is fine, but I don't know, right? I wasn't there. I didn't test all their reagents, etc., etc. Or maybe obelisks are free living. Maybe they favor protists. Maybe, again, maybe bacteria are the wrong place to look. And maybe, as Chad has said, maybe they have a way to avoid getting CRISPR'd, which would be crazy. That would be amazingly interesting on its own. And that would be dope. Or it could be a combination of these things, right? It is interesting to think about, but it is unexpected. It is unexpected. Okay, so in conclusion, with the help of Resta, because before I didn't really have a way to critically engage with this paper, I've realized that including this paper, I'm not going to call it a mistake, because again, reading for pleasure and reading to make a video is a little different. I treat it differently. I'm more reading for fun and to make it fun for you all to listen to and to see on screen. But when I read a paper for stream, and again, I'm sure many of you are new to streams, you get this kind of deep reflective thought, this critical eye, where I am trying my hardest to determine whether or not the paper can be trusted and to what degree the paper can be trusted. So if you're interested in this kind of stuff, make sure to tune into more of my live streams where I do just that, right? I'm actually very curious. I'm sorry to like break up the flow, but I am curious how many of you are new to the stream. So let me go ahead and put up a poll. Are you a new stream watcher? Just for my own curiosity, I want to know. Yeah. So the thing is that, yeah, they couldn't find obelisks in the CRISPR segments of these bacteria. They didn't have VNom work, even though VNom made their, you know, the original paper that discovered obelisks got VNom to work. I don't know why these guys couldn't get VNom to work. And maybe there's a good reason, but they didn't provide the reason. So I have to be a little suspicious there. And then here's a point about thresholds. So Resta, do you want to talk through why or what dimension of a paper can have shaky results due to this concept of thresholds?
Thresholds of...
Oh, just like when you do analysis, because I know before stream, in the short time that we had to prepare, just the two of us, you were talking about how thresholds can really make or break a certain paper.
Right. So when they used Pebble Scout, when they used Pebble Scout to search for putative obelisks, I found it interesting that they used the default settings and didn't try to modulate the actual threshold for like how similar the input sequence is to whatever is in the database. Because if you adjust that threshold of, in this case, Kamer similarity or something like that, you might have like more conservative results or more like wider net results. And so the fact that they found putative obelisks all around the globe makes me wonder that if they made that threshold more stringent, would that pattern disappear?
Yeah, and I'll ask you, because I don't have any experience with this, but how common is it in bioinformatics to just go with the default settings? Like maybe they were like, okay, default settings are good enough, we push it, we go. But is it like really unusual that they just went with the default stuff? Or what okay, so this might be a little heady, and you can feel free to be like, it's a bit much for this stream. But like, is there a simple explanation for why you might choose to screw with those initial settings?
Yes, definitely. As I mentioned before, the default settings can often have assumptions about your data that aren't necessarily true. I was trying to think of an example, but like, for example, if you're looking more so for like modified nucleotides within your sequence, then like just using the default ATCG isn't going to cut it.
Gotcha.
A different program.
So to try to like And you can add on more, but as I'm like digesting what you're telling me, is that like not every data set is made the same way. Some of these data sets have attributes that might be more or less different, and it would be inappropriate to use the same hammer for every situation.
Right. But yeah, typically people who just stick with the default settings don't have as much bioinformatics experience because, like, again, if the authors like were more bioinformaticians, I imagine that they like construct their own pipeline to do these analysis steps as opposed to like using Pebble Scout, which is an online service that like it's just a web page you go to and input sequences. But yeah.
So it's a little suspicious then that they just went for the original stuff.
Like it still works. Like the tools that are made more accessible, I think that's awesome. Because otherwise analyses that wouldn't have been possible for certain research groups are now made possible through the creation of these websites. But the impression I got from this paper is that there was room for more in-depth bioinformatics analysis that I didn't see a whole lot of.
I like the way that you worded that because, again, a big part of my channel is teaching people how to engage critically with science. And I think I like the way that you worded it. There's room for more bioinformatics, right? You didn't say, oh, bad or trash, because so much discourse online is rooted in a lot of critical negativity. And we should never be rooting against scientists who are trying to do the right thing, right? It might not be a great paper. I think it's missing a lot to be a good paper, to be honest, but we're all on the same team, right? And I just wish it were a little better. And I think wording it in a way where there is more to be explored here, that is like moral neutral, right? You're not making a judgment. You're just making an observation that there is more to plumb here, and I think that is accurate. I want to address some of the questions that chat had real quick. Some of the hypotheses that chat has said are that maybe they couldn't find obelisks because they only survive at cold temperature, or maybe when they're heated up, they disappear. The thing is that bacterial memory is rooted in DNA, and DNA is very stable no matter what temperature you're at, unless you're going up like 100 degrees or something, right? But if you're not that hot, then DNA should be quite stable, and it shouldn't matter that you removed the bacteria from their associated ocean layer. And I'm assuming also that the bacteria survive the trip from the CTD all the way up to the boat as well. That's also something that I didn't really bother to think about, but I'm assuming they have methods to preserve the bacteria, at least for long enough to actually process them. Because you can add salts and stuff. If you're worried about DNA melting, you can add salt. Salt will help stabilize DNA. A whole bunch of other things that I don't quite remember, but I wouldn't pick that part of the paper to be critical of personally.
So, yeah. What often, like if you have a CTD rosette, oftentimes they'll have these bottles called Niskin bottles, which are kind of like bottles that have a trap door. So you'll lower it to a certain depth and then close the trap door and then bring it back up. And then once that water is back onto the ship, you'll pass it through filters, again, of a specified size. Because, like, for example, if you're interested in pico eukaryotes, then we'll use filters that can capture pico eukaryotes. And then that filter itself is like a little circle that you can like fold taco shape, put in an Eppendorf tube, add like preservatives of some sort, and then put in a freezer.
Yeah. So there are methods, I think, to ensure the survival of these species. But yeah, again, these two unexpected results, right? You couldn't find them in the CRISPR sequences of the bacteria, and we already know we can find obelisks in other bacteria, so it's a little suspicious that we can't find them in the ocean bacteria, although that could be real, right? It could be that they prefer plankton or something. It just makes my trust in the paper a little less, and the fact that they don't explain these, it's just makes it a little gray. I'm not saying they're wrong, but I am saying if they are wrong, I'm maybe not that surprised. That's the best way to put that. If they are wrong, I'm not going to be shocked. Okay. So, okay, we already did that. This is a different old slide. Okay, that's it. Let's go ahead and let me hide this for a sec so there aren't flashing things on the screen, and I will just hang out on what's a good slide to hang out on? We'll hang out on this slide, this obelisk slide. That's it. That's the entire presentation. I hope you enjoyed that. Resta, do you have any additional thoughts to conclude this reading of the paper or learning about obelisks in general?
So in the original paper that kind of brought obelisks out to the world, one of the things that they did to like prove obelisks exist and are not just like bioinformatic artifact was that they had time series data from the human microbiome. So like the same person over time would have like the same pattern of obelisks within their gut microbiome. And I know that there's lots of time series data for ocean samples. So I'd be very curious to see if the same obelisk patterns are observed across a time series.
That would be really, really cool. I love time series stuff. It can really help open up dimensions of thought that you don't get from just a screenshot of what life is like. More just ask, are biologically active molecules sensitive to pressure differences? I think that they can be. It's just that most of the life that I'm aware of doesn't happen at crazy pressures. So I can't name a pressure sensitive protein, but in theory, it's got to be yes, right? They're just little Legos. And if you can pressurize a Lego, then certainly to some degree, they have to be pressure sensitive. But I don't know that for a fact. That is my guess, to be honest. Yeah. So, okay, before we just chat, I want to make a plug before we just head into just a casual chatting for a couple minutes. This week on the 24th, I will be holding a stream where I just yap, and essentially I will be drawing a unique cell for every one of my members. We got a lot of members from the giant virus and the tiny virus video, and I made the commitment to do that before those videos really took off. But I am still very happy and very appreciative to everybody who has supported me in general. You know, if you don't have money to subscribe, that's totally fine. But I wanted to do an extra special thank you to those of you who have determined that my content is worth a little bit of money. Yeah, words cannot express, you know, looking back on this past year of full-time YouTube ship, like, you know, I went from really worrying whether or not I could do YouTube full time to, for the first time, being optimistic that I could make it happen. Because, you know, my audience knows that I'm a pretty confident person in general, but after a couple of months where the numbers were like, whatever, the continued support from the members and just putting out more content and getting better, I am really starting to see a future in which I can actually do this full time. And I, you know, I thank you all for watching these videos and continuing to be a part of the community. And then a double thank you for those of you who have joined membership. I will say my thank yous properly on the 24th, Christmas Eve. And then the other plug that I have to make is on the 10th of January, I will be holding a 24-hour live stream where I will be raising money for the Breast Cancer Research Foundation. This is a charity that will convert your donated dollars into actual research money, which is incredible. And because you have to have a funding statement, we're actually going to be looking at a lot of papers that the Breast Cancer Research Foundation has funded directly. So you can actually see the impact of where your charity dollars go. Because I think for me personally, it's a little hard to convert, like when you donate to a charity, sometimes you don't really see where that money goes, which is kind of a little sad, right? It's a little abstract. But for the Breast Cancer Research Foundation, I can pull up paper. I could have done that for St. Jude's too, by the way, but I just didn't think about it last year. But this year, I made it a special point to have about half of the papers be funded by the very research money that we are going to try to raise together to combat breast cancer, not only for treatments, but also to improve patient quality of life, to study breast cancer, basic mechanisms of breast cancer biology. Like if there's if you are interested in medicine at all or the biology behind cancer, it's basically 24 hours of me talking about cancer with a couple of special guests who are going to come and keep me company. Like, I'm going to do a Nobel Prize in physiology and medicine tier list with Clockwork, friend of the show Clockwork. I'm going to be doing a marine versus molecular trivia thing with the octopus lady, and if I lose her marine trivia questions, I'm gonna have to pay to the charity, right? There's gonna be a lot of fun, and of course there'll be appearances by all sorts of different VTubers that I have been so fortunate to come across, although I will be streaming because it is 24 hours. It might be kind of hard for all the VTubers that I wanted to invite to come on. Of course, Resta, you are invited to come on. I don't know how friendly that time zone is to you, but of course you are welcome to come and hang out. I think I have it for like 3 a.m. my time because it's 24 hours. That space I have like for my VTuber friends to just keep me company as I spiral into madness. Because I like last year I did 12 hours talking about glioblastoma, and I actually thought 12 hours not that bad. 24 hours seems like a lot, and I'm a little worried that I might fall asleep during streams. So I've invited my VTuber friends to try to keep me up in the dead of night so I can finish the charity strong. So that is my plug for that. And now let's just chat in general. Been a while because I've been on vacation. Resta, you're more than welcome to hang out and chat here too. Maybe about what have you what you have going on, which I've been a big fan of. Actually, yeah, let's take a little bit of time before I go to chat to just, why don't you go ahead and plug what you've been doing, because I find it very, very interesting.
Oh man. So these past couple of months, I'm going back in time. Last week I did a 48-hour film jam with the filmmakers of VR Chats. Crazy amount of time to make a short film. Then the month before that, I directed and produced the finale of a 3D VTuber concert for the agency I work for. Specifically their talent, Leia Memoria of the Seraphine. Then the month before that, I directed and produced a birthday event for their also variants project, but their talent Kacha Sable, which was kind of like mixed between like Paper Mario-esque visual novel plus an animated climax where that shows Kacha being very badass. The month before that, I made an interactive map for variants, like because like VTubers have lore, right? But variants approach to VTubers is like, oh, what if we took that further and really went like very D&D style and kind of created like a cinematic universe of VTubers. I mean, yeah, I guess kind of cinematic because I have made short animation film like things for it. But also in that month, I participated in the month-long August film jam for VR Chat. Had a lot of fight choreography there.
Yeah, I'd say
that was the hardest thing I've ever done.
It's like really cool what you guys, because again, like thinking about our like journey in VTubing, like I remember when you were like messing around with Blender. Like I remember back when I didn't know how to do Blender and I watched your donut tutorial video and I was like, that's cool. And then was that the first time you had any 3D modeling experience? Was that Blender stream?
Nope. Like when I was little, I wanted to make like a paper like origami type figure of like this fox character I liked. And so I put a 2D picture of them into Blender and then like made a low poly model version of that and then put it into another program which converted that low poly model into something that you can actually fold out of paper.
Yeah, I mean that's super cool. Like in terms of like no big secret, if you've watched my videos, I'm like a big HoloLive fan, right? Or at least I was until I stopped watching. I stopped watching HoloLive because I think since becoming a VTuber myself, I've had a lot more fun watching indies than I have HoloLive. HoloLive still has like a very unique place in my heart. Of course, it inspired me to do VTubing to begin with, but I found myself more drawn to the stories of indies these days. But what is always going to be impressive to me is how much Hololive poured into the 3D production. And it's kind of wild that we have here someone who actually does that kind of 3D asset production for an actual VTuber group. And, you know, it's never occurred to me that someone had to make those assets. Someone had to choreograph those concerts. Someone had to set up shots and make sure they look good. And it's been really cool to like follow your journey doing work for variants. And it is like, if I were to describe it in slightly different words, like if you like the big production aspects of VTubing, then I think you're going to get a kick out of what Resta and the rest of the variants project are doing. I think it's super neat.
Thank you so much, Vi.
We have a question from Seed about how do you get full body tracking on your model?
Okay, so there are a couple approaches to that. Kind of like the most common and cheapest one, which can still produce great results, is using IMU trackers. So essentially, you'll have like bands of your arms that you stick these little trackers on, and inside the trackers are IMU chips, that is inertial measurement unit chips. They contain like a gyroscope, sometimes a magnetometer, though typically you want that turned off because like being near a computer is not a magnetically neutral environment. Gyroscope, accelerometer as well. And those provide rotational data. So like as I'm moving my arms here, I have a program called the Slime VR server, which shows the tracker itself, like turning on a particular axis. So yeah, there's IMU-based tracking. There's also lighthouse-based tracking, where if you've heard of Vive trackers, like for VR, you set up a bunch of lighthouses, so to speak, in your room. They're essentially boxes that shoot infrared lasers throughout your entire room. And then they reflect on the trackers and then get sent back to the box. And then it does some kind of calculation. And then from that actually gives both rotational and positional tracking. There's other methods too, like optical-based tracking, either with a webcam or something like a Vicom setup, where you have a whole like rig in your room where you have an array of cameras and you wear the Hollywood suit with all the white balls on them. I know one VTuber that does that, and their studio is crazy decked out. Yeah, there's that. There's now LiDAR-based tracking.
Oh my God, LiDAR.
Yes. There's a company called Movin that has a LiDAR-based tracking system, which I saw they advertised it at a special VTuber mixer in San Diego, California, around the same time as TwitchCon. I believe the event was called Midnight Masquerade. Yeah, there's that. There's also a new kid to the block called FlexPose, which actually uses magnetic fields, but like, because computers, like a typical streamer setup, is not a magnetically neutral environment, they have like this puck that produces its own magnetic field, and the trackers track themselves with reference to the specific field that the puck is producing to isolate it, like the noise from everything else. Because otherwise, like if you don't have a setup like that, even just having like metal like rebar in your floor or walls or whatever can mess up magnetometers. That one currently requires a headset. I hope one day does not require a headset, because it's actually like looking very promising in terms of having both rotational and positional tracking, as well as high fidelity and no risk of occlusion either. Because with the lighthouses, they're shooting lasers, but if the path of the laser to the tracker is occluded, then the tracker loses its position and rotation.
And am I correct to say that you do talk quite a bit about the behind the scenes of VTubing tech? If you're interested, you can go check her out and subscribe to her on Twitch. I also saw someone said high tech VTubing. Are you able to put on your like gear? Your gear is super cool. Are you able to show that off?
Oh, How long are you planning to stream for?
Oh,
I can definitely put on more trackers.
Oh, it does. It requires more. It's not like a toggle.
Oh no, I just have upper arm, lower arm, and hand, and then fingies right now.
Resta has like a whole like
no
mecha
positional tracking either, so
like a mecha suit. I don't even know how to describe it, but like it's super neat.
I've been just kind of delving into all these things, from like motion capture to filmmaking to improv to like fight and dance choreography, and it's been an interesting experience, like really learning about entertainment, even though my formal background is in STEM. Like,
yeah, I only chose to do biology even now. Like, I think that's all super duper impressive. Yeah. So go check her out if you want to hear more about these 3D adventures as well as other related things. Yeah, very cool. I think that's probably. I am feeling a bit tired. I am actually like, I just came back from vacation. Here's a little bit of a yap before I call it a stream. I just got back from vacation, and the thing about me and taking time off is that I am not capable of not thinking about this job. I think if you want an insight into like the mind of a content creator, I think like if you have a 9 to 5 that allows you to go home and not have to think about it anymore, more power to you. On vacation, I loved being on vacation. It was great. But in moments of where there's like no stimulation, I was like, I cannot wait to get back to my computer and make some content. I actually already made a whole 5-bit that I'm waiting to publish about the evolution of the prion protein. So 5-bits are little videos that are supplements to main videos because for some reason or another I can't fit everything I want to talk about in the same video, and I have an update to the prion video, not about the treatment, the clinical trial for the prion protein, but there were some interesting comments about evolution that I wanted to address. I was like, I cannot wait to like do all of the content again and prepare for that charity stream and make the evolution of life video. Like I truly love doing this so much. Yeah. All right. With that in mind, I will hopefully see you for some drawing. I don't know how to draw. Most of the art done on their channel is done by Toxo, so you get to suffer through my terrible art skills, but I do want to make every member a little unique cell as a way to thank you all, and then I hopefully will see you all in the brutal 24-hour live stream to raise money for the Breast Cancer Awareness Foundation. I hope to see you all there. Say goodbye, everybody. Bye-bye. Say bye to Resta. Bye-bye.
bye-bye.
Bye, y'all. Bye-bye.